Good evening, then. I'm going to use this time to go over the most commonly missed questions from our practical and lab. Now, I do want to say that I tried to catch every flag that the system gave and accept as many alternative answers as I could, but I'm going to talk about why I could not accept some answers as being correct. Now, the averages in this class were about an 80 for my biology 1406, that's majors, and right around 70 for the non-majors. Now, if there were students who never took the practical, their grades went in as a 0, so the grade that you see might actually be lower than what the true average for the students that took the test was. I don't want to sway y'all the wrong way and say, well, everybody failed. Well, no, there were 5 zeros calculated into the score, so the average looks lower than it should have been. But that does bring up the question of academic integrity and how I used QuizLog audit. Now, this time, I did not report anyone for cheating, but it was very obvious to me that there was some dishonesty going on. Now, if you see there on the right side of the screen, I have sampled out some of one student's test. And what you see here is the times listed on this side. And it tells me exactly what time you entered the question or the answer for question 24. And then that they stopped viewing the Canvas taking quiz, resumed, and back and forth. Now, when I see somebody leaving and coming back within the same second, I'm going to assume that's a system error and not a problem. But if I see that somebody bounced back and forth this many times, and the time that it took, it were right before 31 minutes here, and they didn't answer until 34 minutes and 40 seconds, that tells me that they went and looked up the answer. And the fact that I can also go in and see exactly how words were typed. I can see when you type 3 letters and then go back, check your spelling, come back, type in a couple more letters, and go back and forth. I'm not dumb, guys. I know what you're doing. And because of this, we are actually going to have to use proctored exams for practical 2. Now, don't get me wrong, many people were completely honest when taking this, and I really appreciate that. But this is also coming up on my quizzes, and that means things are going to have to change. I had some people complain, but I didn't have enough time. Yes, you did. You had more than a minute per question, and there wasn't a single question that required more than 1 to 3 words to answer. There were some that required some calculation, so all right, that might have taken a few more seconds, but there is no need to type out complete sentences on your quizzes or on practicals like this. If I ask you what is the independent variable and your answer is protein concentration, just type protein concentration. You do not need to type out the independent variable was protein concentration. That is a waste of your time. Now, moving on to the actual missed questions. So for Unit 1, this was our metric measurement section. I'm going to be showing y'all some pages that I have taken these pictures either directly from my lab lectures or from our lab manual to show you that all of these questions were given to you in some form. I mean, all of the information was given to you, not necessarily the questions. So it bothered me how many people still have trouble knowing what the word prefix means. And there were a couple different versions of the questions. I can't show you the direct questions because the questions are probably going to be used on future practicals. But if I ask you for the prefix for 1,000 times more than the base unit, your answer should have been kilo, just kilo. It then could be combined to say kilogram or kilometer, but the prefix is just this portion. Now, some people seem to make the mistake of knowing what 1,000 times, and then if another person had the question of 1/1000th, 1 over 1,000, the answer then be milli. But there were other questions regarding 10 to the 6th, for example, that would be micro. So I ask you to memorize this chart and at the very beginning of our course, and I told you it was going to be coming back. So learn it, please, please, please. The next thing that was missed was scientific notation. Exponents matter. If you see here that this first equation is 10 to the 3rd, but the next one is 10 to the -3, notice that that means you're moving the decimal in opposite directions. 3,270 is very different from 0.00327. So if you lost your negative sign, you gave me the complete wrong answer. I can't go, oh, well, they were really close and give partial credit. Your scientific notation should always be formatted a 1's digit, a decimal, and 2 more decimal places, times 10 to whatever you want up here. Now, I was lenient on if you just wrote times 10 to, if it was to the 5th, if you gave me, however you typed it out to give me the inclination that you knew that there was going to be an exponent, if you use a little up arrow, if you put it in parentheses, if you actually typed out the words to the power of, I mean, any of those are totally fine. So as long as you had your decimals in the right place and that you had the correct sign on your exponent. Now, laboratory equipment was missed by many people. 2 example ones here that were commonly confused are the difference between a beaker and an Erlenmeyer flask. Beakers go straight up and down. They don't become more narrow, where Erlenmeyer flasks do become more narrow. For Unit 2 now, for spectrophotometry, I had several questions on there that were talking about which color of the electromagnetic spectrum had the highest energy and which ones had the lowest energy. So I want you to pay attention to the wavelengths being shown here. If the ripples are very close together, that means it's high energy. As energy is lost, the ripples get further and further apart. So that means that the purple end of the spectrum should be the one that has high energy. Remember our ultraviolet UV rave memory mnemonic, and then red being down here at the infrared lazy microwave and has low energy. You see increasing energy, the arrow points up that way. For serial dilutions, you were given a question similar to this, and it showed how much was carried over on each step. So for an example here, if this first tube was 1,000 milligrams per milliliter, and then 1 mil of that got moved over and mixed in on the next tube, now our concentration has been cut in half. If we move over again, it gets cut in half again, and again, and again. Many people miss this question because they weren't paying attention to the tube numbers. It told you what the concentration was at tube 1, but I don't think that it was counting for stock, and some people did not do the translations correctly from going from a stock bottle into tube 1, and then tube 3 is further down the line. So it threw off your steps along the way. Oh, that does remind me now while it's on here. Blank, in this case, it shows water. Your blank only should contain water and biuret. There was no protein in the blank tube. Unit 3 was discussing acids, bases, and pH. So there were a couple variations on questions about anthocyanins and or cabbage juice, whatever you want to call it. You'd have a picture of a test tube, and it would ask you to identify whether it was acidic, neutral, or basic, with acidic being more on the red-pink end of the spectrum, neutral is more in the purple range, and basic gets more and more green, or blue. I remember blue with basic because the B's matched. The other questions ask you what was mixed with it to indicate the pH of the solution in the tube. And your answer should have been anthocyanins or cabbage extract. Now, you can't tell me buffer 4 because I already told you that there was some kind of buffer in the tube. I needed you to tell me what got mixed with the buffer that showed me the color change. So I had to be a little picky on the answers. If you were very specific to say that it was an acidic buffer, I gave partial credit. Or if you said, buffer pH 4, something like that, that showed me that you clearly understood what was going on in the experiment. But you have to read the questions more carefully. I cannot give full credit when you don't answer the question that's asked. There was another similar question to this that had a couple different curves and asked either what was the strongest buffer or what was the weakest buffer. Remember that buffer means ability to resist change. So in this case, the yellow solid line is the weaker one, because if you start adding either the HCl or the NaOH, the pH changes very rapidly. By comparison, the green dotted line there is a stronger buffer because it could resist change and stayed closer to its starting point. Unit 4 was about macromolecules. Now I'm going to take this time to jump over and show y'all the lab resources tabs, because all of the photos that you could want from labs are shown on here. Grab that real quick. Okay, so I am on the Biology 1406-1408 Lab Resources section of Canvas. I showed y'all this at the beginning of the semester. I also posted an announcement to remind y'all that the resource was there. But yet again, here we are, going to modules, and you can scroll down and go through everything. Now, if you'll notice, there are videos, there are images. This lab manual is not the one we use for our class, but I want to show you macromolecule resources here. Now, on this page, you have photos of all of the test tubes. For example, this one was for reducing sugars, with this being negative, slight positive, and very positive. Iodine down here, that is negative and positive. Our protein test for negative and positive, and explanations about the Sudan-4 test here. So you should have seen these test tubes in class. You should have taken your own pictures, but even if you didn't, you had these available for you to be reminded. I told you that there were going to be test tubes out on the tables, and you were going to have to be able to tell what was in the test tubes. That was given to you. For Unit 5, from microscopy, the first things I want to talk about are the parts of the microscope components, because again, I ran into this odd question, or I guess odd answer on many students' pages that gave me the indication that they weren't being honest on where you were coming up with your answers. For example, here, this is pointed in the arrow, or pointed out by the arrow, the word mechanical stage arm. This picture over here to the left is taken from the lab resources page. And then these were photos that were given both in my lecture PowerPoints and in your lab manual. So we can see things like stage and ocular lens, those sorts of things are there. But we always use the phrasing mechanical stage arm. So when somebody tells me it's stage control, I know there's a lot of resources online that call the knob, again, that's this mechanical stage arm is shown here. It's on here and under here, but it was explained in class what it is. We never once referred to this as stage control in any of your study resources for class. So with so many people typing in stage control as their answer, I'm gonna have to guess that y'all exited the quiz and went and looked it up. Not cool. Now, if you kind of gave me more of an explanation of what it did, I might have given you partial credit. And I probably gave partial credit for stage control, but unless you can show me in your notes where you wrote it down, or if it was in your textbook or something as stage control, I can't accept that for full credit. Stop cheating on your quizzes and your practicals. Again, these are pictures taken straight from the lab resources page that were already even labeled for you so that you could practice looking at these. These are things you should have looked at in class. They were also shown to you on quizzes. So the types of structures you should have been looking for, in this case, things like nucleus and cell wall, because this one is a picture from an onion. For the Elodia, the green circles floating around are chloroplasts, and again, there's a cell wall. Plants have cell walls. Human cells have plasma membranes or cell membranes. They do not have cell walls. Our cells are squishy and can pop. No cell walls. But here, the big circle is the nucleus. On the Elodia, you cannot tell what is the nucleus. Green circles mean that there is chlorophyll present, which means it has to be the chloroplast. Your nucleus wouldn't be green because there shouldn't be chlorophyll and or chloroplast structures inside the nucleus. So moving forward, for Unit 6, prokaryotes. A lot of people miss questions related to morphology. Remember, morphology means shape. And so if I gave you a picture of a gram stain and I had said, identify the cell morphology and gram stain results, you should have been telling me either bacilli, cocci, or spirilla. If you could tell me staphylo or strepto, that would be nice. I did also accept a diplo on one because it was hard to see that they were chains that were actually longer than just two, but I understand the confusion on that. So I did accept it, but I really needed at least these base words here. And to talk about gram staining, a purple picture would have been positive and a pinkish red picture was negative. Now there was a question about what do gram negative cells possess that gram positive cells lack? You should have told me that it had either an extra cell layer, extra lipid membrane. Now, you can't tell me cell wall. You might say extra cell wall, but see, there is a peptidoglycan cell wall on both of these. You have to tell me that it has an additional. Both have a plasma membrane, so you can't just say it's a plasma membrane. You have to tell me what is different about them. Gram negative have an extra layer. I would handle you calling it extra lipid membrane or even extra cell wall, even though it's technically not a second cell wall. But you had to tell me something about it. Gram-positive cells do not lack a cell wall. They have a thick cell wall compared to having multiple thin layers on the gram-negative. People also missed the question about what orders the stains go in. We should have started with crystal violet. Then you do iodine. The decolorizer is alcohol or ethanol. And the counter stain was saffronin. Overall, I am proud of how you did on the test, but I found that most of the students who failed were also the ones that were exiting the quiz logs very frequently. So that tells me that you don't have time to go look up the answers. You need to know these answers from your own memory. On Practical 2, you are going to be videoed while you're taking the test to make sure that you aren't bouncing around onto multiple screens. So I'm sorry that it had to come to this, but unfortunately, this is what happens when we can't proctor the exam in the testing center. Now, if campus does open up before your test date, I'd love to be able to have you be able to go to campus and take the quiz on one of the computers there, or not the quiz, the practical. But if you had any other questions that I did not go over and you want to know the answers to, please send me a message and I will get back to you. Thanks, guys.